Professor Tiginyanu and your colleagues, it gives me very great pleasure to talk to you today and to explain the importance of basic research for biomedicine. One often thinks that when you need biomedical applications, you really should focus your research on them. But I hope to convince you during this talk that that is not the case. You can certainly make progress that way, but very often the real breakthroughs come from discoveries from basic research that lead to novel ways of asking questions and answering questions that are relevant to biomedicine. I thought the best way to do that was to give you some examples from my own experience as a basic researcher. In 1970, an important enzyme was discovered by Professor Hamilton Smith, who at the time was working at Johns Hopkins University in Baltimore. And he discovered an enzyme that was actually able to cut DNA at very specific sequences, and it was called a restriction enzyme. He wasn't actually looking for it, he was really looking for something totally different, but nevertheless, this was what he found. And once he found it, a neighbor of his, Daniel Nathans, who eventually shared the Nobel Prize with Ham Smith, realized what it might be useful for. And he showed that one could take SV40 DNA, cut it with this enzyme that Ham Smith had found, and it would produce nice, discrete fragments from this rather large virus. It gave you a way of taking a very large DNA sequence and making it a small one. In 1972, I moved from Harvard down to Cold Spring Harbor Laboratory, and I immediately started working to look for more enzymes like Ham Smith's. And the bottom line was that I was able to find more than 30 such enzymes, all recognizing different DNA sequences rather quickly. In fact, everywhere we looked, we found these enzymes. And we now know that something like 90% of all bacteria have enzymes that are similar to the one that Ham Smith found. Now, these restriction enzymes are incredibly useful, but not only that, they led to some very important discoveries in biomedicine eventually. However, to start off with, they were not commercially available, and so I was lucky that I met a gentleman called Don Combe, who had started a small company up in Boston that he intended to make reagents that would be useful for molecular biology. He had the idea that Ham Smith's enzyme could be very useful, but when he talked to me and discovered that we had more than 30 of these available, he decided that he was going to focus on these restriction enzymes as the reagent he would sell first. The result of all of that was a company started in 1975 called New England Biolabs, and that company has now grown to more than 600 employees. And among other things, we were among the companies that were making important enzymes necessary for the production of the COVID vaccines, for instance. We also make reagents that are very important for diagnostics in biomedicine. We have worked to develop assays for filarial diseases that affect populations in Africa, as well as many other kinds of things. So out of this grew not only our company, but the whole biotechnology industry, which, as I'm sure you know, is now incredibly important in generating reagents that are useful for biomedicine. The second example I would mention is DNA sequencing. DNA sequencing was a basic technique that was developed by Fred Sanger and by Wally Gilbert. And Fred Sanger's method eventually became the one that was widely used and was the one that I was using when I was first interested in sequencing DNA. And we wanted to sequence DNA in order to find out what genes were present and to try to understand how, in the first instance, small viruses actually worked. That was incredibly interesting to do, and the restriction enzymes also turned out to play an important role there because they allowed people to make small fragments of DNA which could be sequenced fairly easily, and then one could join all of these pieces together and eventually get the sequence for something like SB40 virus, which was about a little over 4,000 nucleotides long. But of course, as soon as we had these sequences, we needed to do two things. One, prior to getting the complete sequence, was to develop computer programs that would put them together and would allow us to assemble them. And the second thing was bioinformatics programs that could look at the sequences of the DNA, work out where the messenger RNA was being made, and then where the genes start, so that we could get the sequence of the protein without actually ever isolating the protein itself. We could just look at the DNA sequence and work out what the protein must be. And this was the birth of bioinformatics, another incredibly important technique that, for instance, allowed the interpretation of the human genome, and these days allows the discovery of mutations in genes that cause disease. Very, very important thing. Now, the next thing that happened in my career is that we had developed software for assembling DNA sequences. We had developed some software for analyzing them, but other groups were doing that too. But at the same time, we were very interested in where the genes started. So you look through a DNA sequence and you can easily, usually, tell where the gene is. And so upstream of your gene, you expect there to be a sequence that will tell you where the RNA polymerase should start copying the gene in order to make the protein. In the course of doing that, I came up with what at the time I thought was an incredibly clever technique to look at the messenger RNAs that were being made by adenovirus. And I thought that by using the technique, which looks specifically at the 5' ends of the message, at the short oligonucleotides at the 5' end, that not only could we look at individual messages, but during the course of an infection, we might look at all of the messages that were being made and see which ones went up and which ones went down. So I had a postdoc in my lab, Richard Gelinas, who was a very talented fellow. He'd been a student at Harvard who I'd met when I'd been a postdoc at Harvard. He'd come and join my lab, and he got started on this project. And what had happened is we had a 2D method of looking at what we thought were going to be all the ends of the adenovirus messenger RNAs. But instead, when Richard did the experiment, what he discovered was that there was just one end that he could find. That is, all of the messenger RNAs in adenovirus seem to have the same starting sequence. How was that possible? Well, we did a lot of work. We spent the best part of a year, first of all, trying to prove to everybody that the results we were getting were not an artifact, but more importantly, to find out what the experiment was telling us. And we learned something really important then. The first was that the initial failure was actually one of the best things that ever happened to us. Because in the process of trying to do a post-mortem and find out why the method had failed, we actually discovered something really interesting. And that turned out to be a phenomenon we now call RNA splicing. The fact that genes in higher organisms, like adenovirus, which is a human virus, the genes are split into pieces. They're not a continuous segment of DNA. Rather, they're one piece here, one piece there, one piece further down. And that the way in which they are transcribed and that the messenger RNA is formed is by first making a transcript of the entire gene and then cutting and splicing it in order to produce a messenger RNA in pretty much the way that a film producer, a film editor, makes a movie. They shoot little scenes and then cut and splice those scenes together to give you the impression of a continuous scene going through. And the nice thing about that was that this was a totally unexpected phenomenon. No one had realized that this might be the way that higher organisms make genes. As soon as we reported it, actually, as soon as I started talking about it in public, everybody who was working in an area related to this discovered that their eukaryotic organism was making messenger RNA exactly the way that adenovirus was, and that the genes in higher organisms were split into pieces, into introns and exons, as we now call them. These words were devised by Wally Gilbert. So that was very good. And not only did that lead to a Nobel Prize for myself, which I shared with Phil Sharp, who had independently made the same discovery at MIT, but more importantly, it was absolutely crucial in interpreting the human genome sequence, for instance. And we now know that there are actually medicines that can be made by changing RNA splicing, by correcting defects in RNA splicing, and very recently, not a cure, but a treatment for spinal muscular atrophy was discovered by an ex-postdoc of mine, Adrian Craner, who has now been working on this for many, many years, and in fact now has a wonderful biomedical application of this discovery that was made in 1977. It took many, many years, from 1977 until the early or late 2010s, before the discovery was turned into a medicine. And I think this is what I want to leave you with, that in all of these cases, we didn't do the experiments in order to help biomedicine. We did the experiments because we were curious about how nature works. And as you make discoveries, it is really very easy to see that the discoveries you make can be used, or that others make, can be used in order to develop cures, in order to really enhance our ability to understand disease and eventually to cure those diseases. So let's make sure we continue to support basic research for biomedicine, that we pay less attention to trying to do research that leads to applications, and let's just take advantage of whatever techniques are available to understand more about nature, and I will guarantee this will lead to advances in biomedicine. So I thank you very much for listening to me, and I hope that you have an excellent conference. Thank you very much.